Inside a human cell, cytosolic ribosomes are generating over 1 billion protein molecules at a rate of 5 to 6 amino acids per second. It is a dense, high-speed assembly line operating constantly to maintain biological function. But this rapid synthesis is intrinsically error-prone. Ribosomes misincorporate an amino acid approximately once every 10,000 operations. Without rigorous surveillance, these errors produce misfolded structures and toxic aggregation, a state known as proteotoxicity. For decades, biologists modeled the defense against this toxicity as a strictly localized maintenance routine. The standard view assumed individual cells managed their own misfolded proteins using isolated sets of chaperones and degradation pathways. Recent research reveals a different architecture. The proteostasis network operates as a highly integrated, organism-wide signaling web, spanning from the nanoscale surveillance of a single ribosome to macroscopic fluid dynamics in the brain. The etiology of neurodegenerative proteinopathy lies in the progressive, systemic collapse of this multiscalar network. This diagram shows a cross-section of a ribosome exit tunnel. Measuring exactly 80 angstroms in length and roughly 10 to 20 in width, this tight confinement forces the nascent peptide chain to form secondary structures, preventing premature folding. When the ribosome encounters problematic mRNA sequences or rare codons, the translation process halts. This stalling immediately activates the ribosome-associated quality control pathway, recruiting ubiquitin molecules to tag the trapped polypeptide chain for destruction before it forms toxic aggregates. Localized structural errors escalate into a coordinated cellular reaction. An overload of misfolded proteins triggers kinases like PERK to phosphorylate a critical translation initiation factor, EIF2-alpha. This specific chemical modification marks the activation of the integrated stress response. The phosphorylated EIF2-alpha binds with extremely high affinity to its exchange factor, EIF2b. This physical sequestration stops the recycling of EIF2-GDP to its active GTP state, breaking the cyclical feedback loop required to sustain translation. This creates a precise paradoxical effect. While global protein synthesis effectively shuts down to reduce the structural burden on the cell, the translation of specific stress response messengers, such as ATF4, is selectively upregulated. A nanoscale co-translational error instantly restructures the cell's entire metabolic priority, shifting its machinery from continuous biosynthesis directly to crisis management. Once proteins exit the ribosome, they rely on molecular chaperones to maintain their required conformations. Rather than acting as passive binding shields, chaperones perform active mechanochemical transformations to drive complex proteins into their native folds. By utilizing single-molecule optical tweezers to physically manipulate individual molecules, researchers can measure the exact dynamics of this folding process. The data reveals how chaperones systematically apply force to resolve short-lived intermediate states that would otherwise cause the protein to clump and aggregate. This diagram illustrates the endoplasmic reticulum, the compartment where approximately 30% of the human proteome is synthesized. Because it produces critical secretory and membrane proteins, the ER utilizes a dedicated triage system featuring 3 distinct vectors: ERADL for the lumen, ERADM for the membrane, and ERADC for the cytosome. These pathways sort misfolded substrates based entirely on the physical location of their defects. Once identified, the defective protein is retrotranslocated into the cytosol and fed directly into the 26S proteasome for targeted degradation. By filtering output through these specific spatial domains, organellar surveillance ensures that toxic structures are physically isolated and dismantled before they enter the secretory pathway and compromise cellular communication. In metazoans, proteostasis functions through cell non-autonomous signaling, where a folding imbalance in one specific tissue dictates protective gene expression in completely separate distal organs. This multi-organ systemic map demonstrates how this operates in the nematode C. elegans. Thermosensory AFD neurons detect thermal stress and immediately release a chemical pulse of serotonin and neuropeptides down into peripheral tissues, such as the intestine and muscles. These neural signals activate the transcription factor HSF1 in the gut and muscle, which upregulates the production of the protective chaperone HSP70 across those peripheral tissues. This architecture scales to human endocrine coordination. Localized mitochondrial stress in an organ like the liver or muscle triggers the release of circulating signaling molecules known as mitokines, including FGF21 and GDF15. These mitokines travel systemically. GDF15 acts as a whole-body stress sensor by binding directly to the GFRAL receptor in the brainstem, modulating the organism's feeding behavior and metabolic homeostasis to compensate for the distant mitochondrial failure. Simultaneously, the hypothalamus processes these systemic stress signals. Through the release of neuropeptides like vasopressin and oxytocin, the brain orchestrates a biphasic adaptation, first mobilizing energy reserves, then driving an anabolic recovery to stabilize mitochondrial function. The organism protects its weakest cellular links by leveraging the vast reach of the nervous and endocrine systems, translating a localized folding error into a highly coordinated community protective response. The final physical failsafe of the proteostasis network operates at the macroscopic scale. The brain relies on a system-wide fluid transport mechanism called the glymphatic system to physically flush out metabolic waste and toxic proteins, like amyloid beta. This clearance is driven by the continuous exchange of cerebrospinal and interstitial fluid. Water channels, known as AQP4, located on the end feet of astrocyte cells, regulate a rhythmic pulsing of fluid that washes through the dense tissue matrix. This AQP4-mediated mechanical flushing requires specific interstitial ion concentrations and fluid dynamics that occur almost exclusively during deep sleep cycles. If this brain-wide fluid transport slows down or depolarizes, the structural debris remains trapped in the tissue. Without physical clearance, all upstream molecular and transcellular safeguards eventually become overwhelmed by the accumulating waste. Aging introduces a shift in this equilibrium. The burden of misfolded proteins increases due to genetic mutations and environmental stressors, while the network's capacity diminishes through factors like reduced arterial pulsatility and AQP4 channel depolarization. This imbalance causes a cascading retrograde failure. Local degradation pathways stall, allowing highly resistant, insoluble protein inclusions, aggregosomes, to form. These massive structures physically rupture the membranes of endosomes and lysosomes. The vesicular rupture spills these toxic inclusions directly into the cell's cytoplasm. The physical blockage paralyzes intracellular transport, ultimately killing the cells and clogging the macroscopic fluid channels of the glymphatic system. Alzheimer's, Parkinson's, and Huntington's diseases share a convergent mechanism of network failure. While each features different hallmark proteins, amyloid, synuclein, and huntingtin, they all exhibit similar patterns of ubiquitin proteasome impairment, chaperone sequestration, and eventual glymphatic failure. The precise identity of the misfolded protein dictates the specific symptoms, but the underlying pathology remains the systemic, transcellular paralysis of the body's entire waste management architecture. For decades, therapeutic research focused heavily on attempting to clear end-stage protein plaques after the structural damage was already done. The new translational strategy shifts focus toward pharmacologically tuning the upstream proteostasis network before the collapse occurs. One of the primary agents of this approach is ISRB. As a potent small molecule modulator of the integrated stress response, ISRB binds specifically to the EIF2B complex, forcing it to detach from the inhibitory EIF2 alpha node and immediately restoring the active biochemical cycle. By chemically reversing this targeted inhibition, ISRB effectively restarts protein synthesis in stressed cells. In animal models, this intervention successfully rescues cognitive deficits linked to age-related decline. Parallel strategies utilize chemical and pharmacological chaperones, such as PUAD and TUDCA. TUDCA modulates local drug bioavailability, directly alleviating stress in the endoplasmic reticulum and stabilizing the unfolded protein response. Restoring proteostasis moves the therapeutic focus toward the stabilization of these underlying biochemical circuits. The objective is to maintain the functional balance of the proteome, even in the presence of age-related stressors.